Hello and welcome to this lecture on the anatomy and physiology. We're going to look at both prokaryotic and eukaryotic cells. This chapter is chapter 4 for my introductory microbiology course taken by my college students. This is part 1 of a 3-part lecture series on cell anatomy and physiology. We'll begin by looking at prokaryotic cells, and then we will look at eukaryotic cells as well. So let's get started. So let's look at these two terms, prokaryotic and eukaryotic. Prokaryote comes from the Greek words for pre-nucleus. It's technically pre-kernel, like a seed kernel, but that doesn't make a whole lot of sense in this context. So prokaryote is referring to pre-nucleus, and so it goes back to evolutionary time scale, and prokaryote is a term used for those group of organisms that were on the scene of life before nuclei existed in cells. And so that's why they're called prokaryotes. Eukaryote means true nucleus. It comes from the Greek words that mean that. And so this would be all the other cells that are out there that possess nuclei. And so, like I said, we'll be looking at prokaryotic first. But let's look at a comparison side by side between prokaryotes and eukaryotes. So nucleic acids not in a membrane. In eukaryote, paired chromosomes within a nuclear membrane. And also with prokaryotes, notice I did not use the word chromosome. They have DNA and they have RNA, but they are not wound up around these proteins that are called histones. And the histones are what the nucleic acids, the DNA, gets wound up around so that it can make these organized structures called chromosomes for mitosis and meiosis, which prokaryotic cells, bacteria, do not carry out. And archaea as well, I should say that. Archaea are also within this group of cells called prokaryotes. So these characteristics apply to that group of organisms called archaea, but in this course we don't really discuss them. We look at bacteria, so for our intents, prokaryote is equivalent to bacterial cells. Prokaryotes are no organelles. You will not find an organelle. And by definition, an organelle is a structure that is a membrane-bound structure that carries out a specific function within a cell. And in prokaryotes, there are no organelles. The closest that you have to an organelle, I suppose, would be a ribosome. Ribosomes have no membrane around them, and they are bundles of RNA and protein. There are two parts to them. One part is made of RNA and another part is made of protein, and they're kind of twisted and tangled together. But as far as organelles go, prokaryotes do not have any, eukaryotes do. They have a number of organelles within them. Cell walls made of peptidoglycan in prokaryotes, and in eukaryotes, there are other than polysaccharides, but predominantly if a eukaryotic cell has a cell wall, its cell wall is going to be composed of polysaccharides in some form. How do they reproduce? Prokaryotes binary fission, eukaryotes through the processes of mitosis in somatic cells or body cells, non-reproductive cells, and meiosis, which is the reproductive process for our gametes or our reproductive cells, egg and sperm cells. Binary fission is a pretty simple reproductive method. There aren't any distinct steps or phases like you would see in mitosis and meiosis. And the reason why is because chromosomes don't need to condense, pair up, duplicate, separate, and all that. They don't go through those steps because they don't have chromosomes. So prokaryotes don't need the process of mitosis or meiosis. So they simply just duplicate their nucleic acid chain. It then is sent to opposite ends of the cell, and then the cell just divides right down the middle. And that's binary fission. Their size and shape, they come in a number of different orientations in terms of their shape. As you can see in these pictures here, these are all the basic shapes of bacteria. Their size, pretty small, definitely microscopic for nearly every species. And as I said in the first chapter when we did an overview of bacteria, there are a couple of species that are macroscopic. You don't need a microscope to see them, but it's an incredibly small number of species that would fit that criterion. So nearly all are microscopic, very small, and they come in a variety of shapes and orientations. How are they arranged? These are the three main ways. They come in pairs. For example, diplococci, these would be pairs of round cells. Clusters, staphyle, that prefix staphyle means clustered. So for example, staphylococci. Or chains, where strept means a chain. Streptococci, a chain of spherical cells, or streptobacilli, which would be a chain of rod-shaped cells. So they come in pairs, clusters, or chains. And sometimes they exist as what's called a tetrad, where you have two pairs, so there are four of them. And sometimes they just have no real arrangement whatsoever. They're just independently living. They have an outer cell wall that's called the glycocalyx, and this glycocalyx is usually sticky. Now, if it's a neatly organized one, meaning if it's tightly bound together, held together by these by amino acids and these other acids called teichoic acids, it's called a capsule. It's called a slime layer if it's unorganized if it's unorganized and loose. And so it kind of fits what it is. Slime layer, it sounds kind of unorganized and loose and slimy. And so these are the two different glycocalyxes you'll find in bacterial cell walls. They have these extracellular carbs, polysaccharides, that allows a cell to attach to surfaces where it can grow, so that it can then, this enables adherence of the cell wall to, say, a mucous membrane, for example, and then they can grow and multiply and divide. And capsules will prevent phagocytosis because they're tightly organized, neatly organized, and so because of their complex, tight structure, many times white blood cell enzymes cannot penetrate the capsule, and so they cannot be phagocytized by white blood cells. So the capsule becomes a very protective mechanism for the bacterial cell. They have flagella, and their flagella are pretty advanced for being these ancient cells. They're made of chains of the protein called flagellin. And it's attached by a protein hook, and it basically acts like a motor within as it's anchored into the cell wall, which is that structure, that part of the flagellum is called the basal body. And it rotates in a corkscrew manner. As you can see in the picture there, it's showing it spinning around in a clockwise direction. So these get anchored tightly. They become incorporated into the cell wall, and they're very important for bacteria because bacteria are almost always in fluid, and so they need flagella to enable them to swim. And so their flagella are pretty advanced, pretty complex structures for these prokaryotic cells. This is just a picture showing you the different arrangements of flagella, where they can have just one flagellum, or they could have a bunch of them all over their cell wall. It just depends on the environment that they live in, the viscosity of the fluid that they're swimming in, and so forth. And so the flagella become their most important means of locomotion, getting through a fluid, getting towards a food source, or getting away from a threat. It's very important for their survival. Now, there are two ways that biologists have classified them in terms of their movement. They rotate their flagella to run, which is a coordinated movement, or to tumble, which is a random movement. So they kind of just tumble around through the fluid that they're living in, not really heading towards something or away from something. So these are the two different basic ways that they move. They're either heading towards something specific, or they're just tumbling through life like some of us live. We kind of tumble along. They move away or toward stimuli, and that's called taxis, like chemotaxis or phototaxis, meaning moving toward or away from a chemical, or moving toward or away from light. And there are H antigens on flagella, these flagellar proteins, and they're useful in distinguishing variations within a species. Like, for example, the E. coli that causes severe diarrhea, some cases dysentery, but not often. It's usually what we associate with what's called traveler's diarrhea. It's the pathogenic strain of E. coli that produces a toxin called the Shiga toxin. You'll notice after its name E. coli, there are these other designations, O157, and then a colon, and then H7. That H is the H antigen. And so we can designate this as the toxin-producing strain of E. coli, not the one that is living in our large intestine, but these are ones that are mutated, and so we can use them as distinguishing variations. So these are slight variations within species of bacteria, and we can use them. And these are found in their flagella. And here is a bacterial cell running and tumbling, tumbling and running, going in a coordinated way or just no real coordinated movement at all. There are other structures that are kind of like flagella, very similar to them. They're called axial filaments. They're endoflagella. Endo means within. And so these flagella are within or deeper down within the bacterial cell. We find them in spirochetes, this is just a group of bacteria with that particular shape called spirochetes, and they're anchored at one end of the cell, and it rotates, and it causes the cell to move in a corkscrew manner. So there are some bacteria that have these, I don't know if you call them maybe like pseudoflagella, that we call axial filaments, that enable them to move all over their body in a corkscrew manner. There are these two other structures here, one called fimbriae, which is plural for that, and pili. So fimbriae allow attachment and colonization. In that picture, you can see the fimbriae are coming out. Imagine them being like roots anchoring the bacterial cell into a surface. They have a few to hundreds per cell. Pili, however, they only have one or two per cell, and they're used to transfer DNA from one cell to another in a process called conjugation. This is not a form of reproduction. It's a form of sharing DNA and nucleic acids. And this is one of the primary ways that bacteria are able to mutate and evolve, through the sharing of DNA. So if the DNA has mutated in one particular bacterial cell, and it links up with other bacterial cells through the process of conjugation, it can share that mutated DNA with other cells, and now they have developed, say, antibiotic resistance, for example. And this is how antibiotic resistance has spread through the bacterial world, through one of the means. It's not the primary, but this is one of the means in which it has spread, through the use of pili, transferring DNA from one cell to a neighboring cell. So as I said, and you notice in this picture from Brie, hundreds per cell in a lot of cases, and think of them as enabling them to root into a surface like a mucous membrane in our throat or intestinal tract, for example. We'll look at the cell wall now. The cell wall gives rigid structure to the cell and prevents it from bursting, osmotic lysis. Bacteria are often in a what's called a hypotonic solution, which we'll look at in a few minutes, and because of that, they need a structure that prevents them from bursting or undergoing cytolysis. If you remove the cell wall, they often do burst because they lose that rigid structure that prevents them from bursting and blowing up. It's made of a carbon-protein complex called peptidoglycan, and it's found nowhere else in the biological world. It's only found in bacteria. And it's a repeating chain of carbohydrates with proteins interspersed within them. So this is what it looks like. There are two disaccharides called NAG and NAM, and they're linked together by polypeptides, which would be amino acids. So think of the cell wall as being like a log raft, where the logs are the carbohydrates, NAG and NAM, and the raft is being held together by ropes. Think of the ropes as these protein chains that are holding them together. That are them meaning the polysaccharides NAG and NAM. So it's holding together these simple carbohydrates, and the proteins are linking them together, holding them together, and giving them strength, and serving as cross bridges between them. Now, looking at the difference between gram-positive and gram-negative bacteria, you'll notice with the gram-positive cell wall, much thicker layer of peptidoglycan. So multiple layers of NAGs and NAMs, NAGs and NAMs, and then you have a little space in between the peptidoglycan, a very small space, and then the inner plasma or cell membrane. So that's gram-positive. Peptidoglycan, thick layer, and then the cell membrane. With gram-negative, you have an outer membrane, then you have a very thin layer of peptidoglycan, and then you have the inner cell membrane, which then within there will be the cytoplasm. The differences between them in terms of, from a clinical aspect, usually much easier to treat gram-positive rather than gram-negative. And the reason why is the gram-negative outer membrane is pretty complex. It's a complex structure and has a variety of compounds within it. And because of its complexity and variety, it's difficult to penetrate. That's one of the reasons why. There are others, but that's the main one. The gram-positive, it's just got the peptidoglycan, and if we can tear the peptidoglycan apart, we can then lead to the lysis of the cell. So even though the gram-positive cell wall is thicker, it's simpler, easier to penetrate, easier for antibiotics to break down, and disinfectants. So look at comparing them side by side. As I said, gram-positive has thick peptidoglycan, gram-negative thin. There are teichoic acids in gram-positives, and those acids are there to maintain the thick structure of the peptidoglycan. Gram-negative doesn't have a thick peptidoglycan, so no teichoic acids. There's no outer membrane in gram-positive. There is in gram-negative. There's a very small or no periplasmic space in gram-positive. That's the space between the the cell wall portion, the peptidoglycan, and the cell membrane. Gram-negative, they have a pretty significant space in which chemical compounds are housed and other things as well. Gram-positive are susceptible to lysozyme. Lysozyme is an enzyme that's produced in abundance in our body. Gram-negative, they are not susceptible to lysozyme. Lysozyme typically is not able to break down gram-negative cell walls. Gram-positives are susceptible to penicillin and sulfonamides. Gram-negatives susceptible to streptomycin and tetracycline. Gram-positives are resistant to lysis because of their thick peptidoglycan, so they're resistant to bursting. However, the peptidoglycan is easy to break down, and so once that happens, they lyse or burst. Gram-negatives are susceptible to lysis because they don't have a thick layer of peptidoglycan. Looking at gram-positives, teichoic acids, as I mentioned, they strengthen and support their thick layer, the multi-layer peptidoglycan. They have a negative charge, and they may regulate the movement of cations, or positively charged ions, through the cell, in and out of the cell. The polysaccharides provide antigenic variation, which enables us then to identify bacteria because of the different antigens that are present within the cell wall.